Hello everyone, welcome to Just Biotech Geeks. I hope you would have watched all the previous tutorials on molecular docking in which I have demonstrated the steps for protein and ligand preparation, setting the grid parameters, and protein-ligand docking. In this tutorial, we are going to see how to visualize a docked protein using Chimera. I would like to mention that I will be providing the link to download all the softwares as well as the websites and commands required for molecular docking in the description of this video. First of all, I would like to demonstrate on how to install Chimera software in your system. You must go to folder where you have downloaded Chimera. Here I have already downloaded Chimera in my computer. Now just double click on the setup file. Click yes. Now click next. Click on I accept the agreement. Click next. Further click next. Here we have an option to create a desktop icon. Leave it as it is. Now click next. Now you must click on install. This installation process might take some time, so please be patient until the process is complete. Now click on finish. Here the software is creating some necessary files for visualization, so please don't close it until the process is complete. Okay, here we have completed the installation process successfully. Now you may close this folder. Next, we will launch Chimera software. Here we can see the Chimera interface. Now let us go to File, click on Open. Now you must select the path or the working folder. Here my working folder, that is molecular docking, is on the desktop, so I will be selecting this particular folder. Now you must select docked_protein.pdb file and click on Open. So here I can see the 3D structure of my protein docked with the ligand. Just for better visualization, I will change the view by clicking on Present and select Interactive 1 ribbon. Now you can even rotate the 3D structure of the protein molecule just by clicking and moving the mouse. Next, you must go to Select, click on Residue and select the second option, that is LIG. The term LIG might change in some case, but you must always select the second option. Now select the option. Here you can see that the ligand molecule is being selected and is highlighted. Now we must check the number of hydrogen bonds between our protein and ligand molecule. For highlighting the ligand molecule, you must go to Tools, click on Structure Analysis, further click on Find H bonds, and now you must select only Find H bonds and select this checkbox for highlighting the bondings with the specific color. Now increase the width of the line by 3 and now you must click on Apply and click OK. You may scroll using the mouse to zoom in and zoom out. So here we can see two hydrogen bonds. The highlighted one is the ligand molecule and the one in red color is the hydrogen bond, and this is the protein molecule. So when we take our cursor near this particular bond, we can see that the ligand is bound with proteins on LYS45 and the next bonding is with GLN46 amino acid. So in case if you want to label this amino acid, press and hold Ctrl on the keyboard and click on this amino acid. Now this particular amino acid is being selected. Next, go to Action, click on Label, click on Residue and select Name plus Specifier. Now this particular amino acid is being labeled. Similarly, you can also label the next amino acid. Here you can also change the font size and style. So go to Action, click on Labels and click on Option. Here you can change the font, but I will keep the same font. You can also change it to bold as well as change the font size. Now click on Save, close this window. Here you can see that the label style has been changed. Next we will be checking the amino acid with the result that we have obtained from Metapocket in part 2 of molecular docking. If this amino acid matches with Metapocket result, we can conclude that the ligand is bonded with the protein molecule at the active site. In case if the result of the amino acid does not matches with the Metapocket result, we can conclude that the ligand has not bonded to the protein in a proper manner or at the active site. So for checking the Metapocket result, go to Google Chrome, search for Metapocket, click on the first website, click on Home. Now enter the PDB ID of the particular protein molecule. In this case, the PDB ID is 2FK4. Now click on Find Pocket to find the active site. This process might take some time, so please be patient until the process is complete. Here I already have the result, so now I will be checking if LYS45 is present in the Metapocket result. Here I can see that LYS45 is present. Next, let's check for GLN46. So both the amino acids are present in the Metapocket result. From this, we can conclude that the ligand has been bonded with the protein on the active sites. You can also view the protein ligand structure in various other formats such as sphere and surface. So here you can also change the background to black or white as per the need. I'll keep it as black. Here you can see the style is being changed to interactive view. Now I will be demonstrating how to save the structure in image format. First of all, position the image in which you can get the best view. Now go to File, click Save Image and select the particular folder where you want to save the image. Next, you may name it as one or any name of your choice. Next, select the file type as PNG or TIFF, which can be used for publication purpose. Now, click on Save and click Yes. Once I go to the working folder, we can see the image file over here. So that's all for this video. Do like, share and subscribe to our channel.